Now our next speaker is Dr. Abhishek Rathore, and he is going to speak on genomic selection population, if I understand, because it's unscheduled presentation, or rather it is a multi-algorithm pipeline for genomic selection. Yeah. So Dr. Rathore, the floor is yours.
Thank you very much. I'm going to present a software pipeline. Well, as Dave said, that we need to use pipeline with much care, which are quite automated or doing the analysis. But this pipeline is to do genomic selection, which is the emerging area. And so we have developed a pipeline which incorporates multiple algorithms for doing genomic selection. So this project or pipeline is called as ISMU 2 version 2.0. Actually, it's an next version of a pipeline what we have developed and last year, which was ISMU 1.0. That pipeline was meant for SNP detection, and where we have the reference data or we do not have even reference. In that case, we can do the SNP mining and then confident SNP, and we have several kind of scores there. Based on that, we can see whether SNPs are confident or not, like read depth. So when that data is available and then we have the phenotypic data available, next step was a very natural step is to use the genomic selection, which is still gaining much momentum. It's a very popular technique. So if I directly come to the genomic selection, there are several forms of the molecular breeding. This start from MABC, MARS, and many other techniques. And then genomic selection is a tool which we say nowadays it can accelerate our breeding cycle because it has several advantages. It incorporates several things, and it increases genetic gain per cycle. Mainly, the most important point is through early selection. It is also, genomic selection is also very useful for the complicated traits. Well, complicated means there can be several kind of things which say complex traits. Sometimes traits may be very expensive to phenotype, may be very difficult to phenotype. Some of them may take very long time to phenotype, and etc. This totally depends upon the breeding program in which we are working or the traits and crop on which we are targeting. So once we have our breeding program defined and we want to work on a particular trait and we think genomic selection is the methodology, we predict breeding values based on that genomic data, and these are called the Genomic Estimated Breeding Values, GEBVs. And instead of using the traditional breeding values what we obtained from the phenotypic experiments, we use the genotypic breeding values which we get by using the marker data. I'll talk more on that. Well, so a few interesting talks have already been here and few more will be. So for that, several analytical approaches or genomic selection models has been proposed for prediction of GEBVs, because as precise our genetic GEBVs will be, better and accurate selection we are doing through the genomic selection. When I talk for the analytical approaches, so in past and in even now, and I know in the future also, daily some new techniques on different crops with different parameters are being tried. And in that, several statistical methods have been developed to handle the genomic selection data. We all understand that in genomic selection data we have few hundred or few thousand lines, and then we have few hundred, thousands, and even sometime in millions of the marker. So this is a problem in which we use all of the marker together in genomic selection, not like GWAS where we use only a marker or a haplotype. Here we try to use whole of the genome marker together. That's why it becomes a high dimensional problem. Like in statistics, then you cannot directly solve such problems well, and we have to use some other kind of techniques. So these high dimensional data analytical techniques are applied in genomic selection. There are several of them which I'll talk in next slides. But the main, I won't say the problem, like when you apply those kind of techniques into biological data, into sequence and the phenotypic data, they sometimes behave different with the different kind of data. And so their respective properties of these algorithms is not very well understood. In some cases it is, but in general if you change the population, it's not very well understood. That's why till date we do not have any very deterministic method to use the genomic selection. In case of some other method like in GWAS, there are at least few very established methods. Still they need to change based on the population and the crop. But here, like in genomic selection, many things are there which we need to consider. If we see the factors affecting genomic selection, I try to just write down a few. They are not the all or the. Sometimes based on the literature, we say that marker density or the genome size and even the population structure or the genomic arrangement can be responsible for the genomic selection and success of the genomic selection model. Sometimes the training population size, if LD is very high, we may because we cannot say, well it's quite logical, if LD is very high we may need to take more markers, but then again how many markers and even more markers, what method. Effective population size, that's also a very important criteria, which model you should choose and what accuracy should come. Trait heritability, there has been several publications from, and many authors are here in this meeting who have worked on that. The trait heritability is also very important in genomic selection. Sometime between relationship between training population and candidate selection, like we have tried in some other environment and then we are predicting for another environment. Will it work? It has worked in few cases. It has not worked in few cases. So those kind of presence of these kind of things are very interesting, and again which kind of model is able to incorporate these kind of fluctuations in our population. Number of genes, distribution and many other things, and again the method what we are using to estimate the marker effect that directly jots down to the statistical model, and there are those things. G by E, which of course is my fourth point, the relationship between training and well, not directly related to that, but it's like we have some other population and another population on which we are trying to do the genomic selection. So there are several factors and they do not give us choice to select only one model. So if I try to write down, so once the first problem comes, what kind of model or what kind of statistical technique we should go ahead with the genomic selection. And once we are sure about that, not really, then we have another few steps. Like in genomic selection, once we are ready with our population, that we need to get a training population. And of course, as in my earlier before the Dave said, that the data filtering, data cleaning is very, very important. What kind of, how should we do? If we really do lots of data filtering or quality control is very important, then there may be chance that we are reducing the data what we are going to use. And if we are taking, then there are problem of over prediction and model misfit may be there. Once we have the quality control, then another thing comes. Are we considering the population structure? Are there some other covariates or weather covariates what we need to consider when we are fitting the multi-environment or even the genomic selection models? Once those things are done, then we need to fit the all available model, whatever like we have, and then perform a cross validation. I think all of understand cross validation, that we take some portion of our data which we use to fit the model. Like here we can divide our data into 5 folds, 5 parts. We made a training set and then we made a testing set, and then this is the way we try to, so this is you perform the cross validation. Then we prepare a matrix of all of these scores and we fit all of the models, and then we select the final model what we is based on all statistics. Then we predict the GEBVs for the data what we have not yet phenotyped, but we have the genotypic data available. We calculate their breeding values and then we select the top model, the top performance there, and then we go ahead with again the making process, get the another population, and then process starts from the top. So this is quite complicated. Well, I said that it's a stepwise process, and there are some more steps in between, but these are the quite common kind of steps. So I'm not saying it's very difficult. My slide may look a bit scary, but it's a quite chain like of interconnected process. You start from one end and then you reach to another end. And if there is a one link broken somewhere in between, it's a whole chain. Like you had a wrong selection of model, your data quality is not good, or even you have not opted for all of the available model in the literature, what may be better suited to your data. In that case, our model fitting or the predictions of GEBVs may not be very confident what we could have achieved. So what are the bottlenecks here? Like it looks like a good flow, but there are few steps already quite clear. Because if we start from the data when we received it from a lab, it starts from raw reads. Well, we can use any pipeline to call the SNPs or the ISMU one you can use to call the SNPs. But then most of the genomic selection models are implemented into R, and many of them are in their other independent languages. Few are in FORTRAN, few are in C, and other languages of choice. People are doing their work and using different. And sometimes when you use those kind of different tools, like you use one type of tool, like you can use Plink to subset the data, or you can use Tashel to subset the data, and then take that data, convert it into another format, and then use some other R, take into R, use the genomic selection, take data out, make some plots, and those kind. It's quite complicated when you start to use this kind of stuff. So we thought that there's a need of a suite of software or pipeline which deals with all of these steps in one without compromising on its power. Like all of the steps should be there, but it shouldn't be so automated, or that automation should not take the power of the genomic selection or the whole process. So that's why we developed ISMU 2. Well, it's also part of integrated breeding platform. It is available through integrated breeding platform website also. And these are the features. I'll show you the pipeline in next few minutes. So these are the features of the pipeline. That it's a GUI. It's not a simple GUI actually. It's a IDE based GUI. It's an integrated development environment where you can use several kind of data sets, you can work, you can save results, you can have population structure, everything at one place. I'll just show you. It has a multi-core support, well not with all of the scripts, what scripts supported multi-core performance, those scripts are here based on the multi-core. So then you can, even if you have a multi-core machine, or even that you can use the multi-core architecture of your machine to crunch the data. Because those who have done the genomic selection, they might know if you want to run 200 cross validation for a very small marker data size like 10,000 markers and 500, that 200 cross validation may take you 10 to 15 days sometime when you run the Bayes and stuff. But I'll come, and then it has a project mode development. You can have several projects developed and then you can save them and you can open those projects accordingly. Multiple method and traits ones. All of the process flow, what I talked earlier, that process flow clearly demonstrated that you can take one trait, two trait, but this pipeline gives you a power that you can use all of the traits all together with all of the methods. And platform, right now we are developing for 4 of the platform. It's a Java based platform on Windows 64-bit, Windows 32-bit, CentOS 64 and Ubuntu 64-bit. The platform is available. And these are the features that it has: the data diagnostics tools, it has graphical summary, tabular summary. We can directly subset data in the pipeline based on the missing percentage of marker. You can review a percentage of missing percentage, minor allele frequency, or the polymorphic information content. The methods it supports are many. Ridge regression blub, the Gaussian implementation of we call kinship Gauss, the ridge regression, Bayesian lasso, Bayes A, Bayes B, Bayes CPI, and machine learning algorithms kind of random forest regression. It supports output in several formats. It supports output in Excel format, in HTML format, and in PDF format. So when you launch the pipeline looks like this. So it looks very simple interface, very few menus. So to start genomic selection, all what one need to have is a genotypic data file and a phenotypic data file. Presently the pipeline supports SNP format where you can have A/T, A by T, or even without separator, or you can have the dominant marker system, 0, 1 kind of data is also supported. Pipeline automatically detects what kind of marker you have given, marker system you have given. You need not to specify a SNP, or it can determine. One need to click on the open, and then you need to specify a genotypic file, a phenotypic file, and a result directory where one need to work. These are the minimum requirement. Well, we need to have a genotypic and phenotypic data, and once we select OK, based on the marker type, it will load the data here. You can see that this is the SNP data what we have given, and it will load the data. The phenotypic data file is here, and the genotypic data file here. You can just see, you can browse. Now if you want to make the summarization of your data file through the ISMU, that you can do, because you may not blindly use a MAF threshold, or you may not blindly use a missing percentage threshold. We may need to have idea about our data. So here is the menu. If you see that the filter menu that says that the genotypic, the data summary. We click on the data summary, and then it asks calculate missing percentage, PIC, minor allele frequency, and if we want to calculate the data summary for our phenotypic data, you want to see its distribution. Well, a pipeline accepts the mean data set. You can give it mean data set, blobs or blues, whatever you have calculated, and then we click on the start. Once we click on the starts, pipeline gives us graphs and the summary table. The graphs shows this, like this graph is a histogram which shows that missing percentage on the x-axis and the total how many marker or the percentage of marker in following in that band is on the y-axis. So this gives us a clear insight of our data. Okay, the 20% missing, I have around 69% of the data which is 20% missing. That kind of, this helps us to provide a cutoff that what if I take a missing percentage 10%, that how many my markers are going to be removed from the further analysis. Similar kind of graphs are available for polymorphic information content and minor allele frequency. All this information is also given in pipeline in a tabular form. So we can browse from here. Once we have this, and if you want to do some more kind of filtering or summary on these markers, directly you can export this file to Excel. Right click here and click on export to Microsoft Excel. Well, you should have Excel there. And so then all of this data directly goes to the Excel. You can save it and you can start to draw the diagrams or whatever you want to sort and make summarization, one can do here. So once we are through with our data quality, we know that what kind of data we have, and same with the phenotypic summary with the histogram, box plot, and if any tentative outliers are there, all those things are plotted there. Here comes the main screen for doing the genomic selection. There is a second menu on the top where we click the data analysis, and then genomic selections dialog box pops up. It's very intuitive, like anybody can understand. They do not require any algorithm or any command line stuff. You say that it is saying that genotypic file name. If you click here, if you have loaded multiple genotype data set, you have a DART data set, you have some other population, all those data sets will be listed here. Select the data set what you desire. Phenotypic file, select data set from here. If you have uploaded any covariate, I have not shown that from where covariate has come, but covariate can be imported later. Select the covariate, and as I selected these two traits, it will show all available traits inside your CSV file. You select any one of trait, or you can select all of the trait together. And now whatever method you are interested in, Bayes regression like Bayes CPI, Bayesian lasso, or we can select all of them. Here we can specify the thresholds, but having a GUI doesn't mean that we cannot really fine tune our analysis. So to fine tune our analysis, we can specify those who understand. Otherwise we have really taken the values which fits most of the data sets. Otherwise one can fine tune these for Bayesian analysis like burn-in, thinning and rounds. If we are using random forest regression, then this will be enabled. It's disabled because we have not selected random. Then we can specify how many forests or those kind of things. We can specify the number of processors we want to use. This slip box will automatically populate to the maximum number of processors available in your machine. If it is 6, it will specify maximum number of 6. So, and then for the cross validation, we can specify the how many replicates in the fold. Click on start. So these three things, and you will get a box like this, and then results are here. So we have all of the results. Now, since we have selected one trait genomic selection analysis is for trait 1, we can scroll down. So this is the histogram of phenotypic trait. Because we generated the output in form of a report. In report, it will have the all phenotypic data summary, the phenotypic histograms. You can see histogram of trait 1, and then here from here start the box plot was on the on above, but will, and then genomic selection random ridge regression R. Like this ridge regression method was used from the R software. The R programming language was used to run this ridge regression. This is a scatter diagram. You can see that genomic estimated breeding values are on this side, and then the observed breeding values. So whatever data set we have provided, it will estimate breeding values for them, and for the values for the genotype for which we have placed only genotypic data, for them it will estimate the GEBVs also. So it do fit as well as the selection together and the prediction together. And it is showing ridge regression method correlation 0.5 and the p-value. So like it's a significant correlation. Well, it was a simulated data. So and then you can use the menu to export all of this output in form of a PDF. So you can say, if you click, then all of your report has been saved into a PDF format. It's ready. Not only that, if we want to use this kind of plots in our publication or in our presentation, so all of these plots are also saved in the folder what we have used, what we have specified during our analysis start. All of these graphics in 300 dpi, these all graphics are being saved in that folder. So on very high resolution. So I have placed from that pipeline also. So you can see they are really on very high resolution. If we go to that folder, you see that all of the analysis, graphics, file, folder, HTML reports are saved there. Whatever reports you want to make into PDF, ISMU will do for you. So you can see that everything is saved, whatever we have done. And support for large data set. That means that if we have really large data sets, large means, well, large is a very relative term. I say large like we have 50,000 markers and 2,000 individuals, maybe a bit large for the genomic selection, but depends upon that like person to person. So here like we have our collaborator John here, John Hickey. He has written some very fast scripts in Fortran, which are the native Fortran scripts. Like for the Linux we have the Linux compilation, for the Windows we have the Windows compilation, and those two kind of 32-bit version have 32-bit compilation. We have embedded those also in the pipeline. So if you think in the primarily analysis it looks that ridge regression and Bayes A are really very good methods, then but we have, as of now we are having two algorithms for that. I'll just show you. So like R is relatively slow. Okay, when I say slow, that means relatively. It depends what is slow and what is fast. So a cocktail of native Fortran binaries and R was used as a solution. When we have smaller data sets, we have choice for more model. In that case we can use the R, and if we have very large data set, then we can use the Fortran, or both. Well, one may take for 40 minutes, another may take few hours. And what we have done, we have used Fortran to generate the results, but the kind of graphics R can produce and the HTML PDF report what R can produce is, I think, a bit difficult to produce in Fortran. So the cocktail has been used, like processing of data processing is being done in R, but the results are being projected into a Java pipeline, and the middleware is Fortran and R. So how to use R? Again, click on the analysis menu. We can select engine from here. When we click engine, it selects either an R engine or Fortran engine. Then we select the engine as the Fortran. And once you click again the same menu, now you get only the two methods. Ridge regression, Bayes A, and you same way, nothing has changed, only this place instead of having 5 or 6, you are getting only 2. Select the traits as usual what we do. Here the cross validation has been implemented. In R it takes lots of time and very complicated, but here the cross validation is implemented. You specify the replication folds and then specify the number of processor, how many you want to use, click on the start, and the same analysis is done by use of the FORTRAN. We can see that again same way here because we have selected cross validation, so now apart from that we are getting a cross validation results also here and then the same are the plots. And like these kind of things, like if we select both of the method, then it gives a tabular summary of all of the methods in the bottom. So you select all traits, all method, click run, sleep. In the morning you may get all data with you, all results in nice tabular form. And well, it gives a time stamping and many other things. So, well, we have the future plans. Like we are going to work that we can make an online kind of version. When I say online, that means web based, because ISMU we have written in 3 layers, so it's very easy, not so easy, but like we have possibility to take the top layer as a web browser and we can do an online version. The one thing which we couldn't implement it, I wanted to import several popular formats, like it should import VCF, PD, BD, or even the HapMap file. We don't have provision for that. For HapMap we are building, but we want that they should have some new methods in the FORTRAN or C which are really run faster. Well, I think we have collaboration with John on that, and we can have some more kind of scripts. Multiple trait genomic selection together, and try to work on G by E. I think we are discussing with Patancheru on that, and so those kind of things we are planning. And there are several institutions with which we have worked in this pipeline development, and thanks to all our colleagues and collaborators. Thank you very much.
Thank you, Dr. Rathore, for that informative presentation. We have time for about 2 to 3 questions, because I would like delay for coffee. Okay, please.
Under global warming conditions, the climate is changing erratically and fast. So under changing climatic conditions, your trained population will be useful for selection.
Training population will be useful?
For changing climate.
Yeah, like this depends, like what population, because any of the statistical method what we used from simple analysis of variance to genomic selection, they all are not the magic. So the kind of training population we have used, same way the prediction population will behave, yes.
Yes, please.
With next generation sequencing, we know that millions of data points per genotype can be created in any case. But do you have some information about the number of marker data points, minimum number of marker data points, which can be used in genomic selection? Have there been any studies using training and testing populations to simulate the number of markers which shall be utilized, minimum number of markers?
Yes, I think there are many papers, even from the RACS and from, I think from the John Luke, where several simulated data sets have been taken, and several methods. I think John also had some paper where simulated data has been used for several, but well, you can do that, and that is the advantage of such tools what we have here, that you can just vary the thresholds of the data quality, and then they will, based on that, the marker number of will really reduce. And then if a GUI, you just click and then you get the result, and then you can, all the table comes at the bottom, and then you can even standardize for your crop. So I think these kind of tools are very useful for those things.
Okay, here first.
Dr. Rathore, from this side, you have given the ISMU to the 2002 version.
Yes.
So can you provide the software of this ISMU to 2.0? Because
yes, indeed we need
that can be useful for the students of
yes,
MSc and
sure,
PG students.
Sure. Yes, we can give you even a CD in a pen drive you can take from us, and it's also part of integrated breeding platform. I think from their website also you can download it. Yes, thank you.
One comment, one question. With respect to climate change, anything that speeds up the breeding cycle helps with climate change adaptation of cropping systems. Farmers are safest who have the varieties that have been bred in the current climate. So genomic selection has a lot of potential to speed breeding cycles and help with climate change adaptation. Farmers in the U.S. Corn Belt who use varieties that took 4 to 5 years to develop and then are in the market for 4 or 5 years after that, very efficient breeding and seed systems, are much better buffered against climate change than farmers who are using 30 to 40-year-old varieties, as many farmers in South Asia are. My question is on your pipeline, on the cross-validation. Cross-validation accuracies are incredibly sensitive to structure in populations. You have a mechanism for controlling for structure within the pipeline?
Yes, the population structure can be provided as a covariate here, like if we have those principal component scores, or if we have very clear-cut population structures, and even other covariates we can provide in the pipeline. The third field for the, so yes, we have option, and that also used during the cross-validation also. That's a very important point, yes, thank you.
The last question over here.
So you said some missing data is permissible. So 2 parts, how much is that permissible, and if it is permissible, so are you going by imputation? And if you are going by imputation, is it acceptable to go for genomic prediction based on that?
Yeah, very good question, actually. So first thing, yes, pipeline, I have not highlighted, but pipeline takes missing data and based on the threshold what you select, and it imputes. So it has 2 imputation method, like automatically it selects. Most of the time we select the nearest neighborhood method, and it takes, and sometimes we replace it with the minor allele. Or we are in process of that, is that what kind of imputation method can be used. But it's a really very tricky, but I think if we have a fair enough coverage of the markers for the genome, and the cross-validation comes good, as Gary pointed out. Cross-validation is very important. Because sometimes if your model is overfitted, you can get correlation value from your training population as 0.9, 0.99 kind of those things can come. But in cross-validation that will totally fail. So if the population structure and missing percentage what we are taking, and it is reflecting in cross-validation, like one of our study, I think Manish, we have 2 Manish, so they have done, you can see the poster outside. In one we have used 400-fold cross-validation because there were some problems. Otherwise usually people use 10 replication and 5-fold, but we have used 400-fold. It took us around 1 or 2 months. There were some reasons. Many experts may say that's not required. And so yes, and then we were very confident about our correlations and our marker profile, what threshold we have taken. We varied in that paper threshold from no missing percentage to 30% missing percentage data allowed. And then we have seen that what kind of results change. You can see that poster, and then I think, so it's a call, like how you do it. That's why again I'll vouch, that's why the pipeline is here. It's very easy. You just click 2 times and your results will change. Yeah? Thank you.
Thank you, Dr. Rathore.
Thank you. Thanks.